Doctor Who is a long-running sci-fi TV show that takes its characters all over the universe and beyond, sometimes leaning into the science and sometimes leaning into the fiction. Finding the line between the two can be difficult, but with some research and scientific analysis, you can uncover the truth. My name is Leo, and I will be explaining the plausibility of a clip from the Doctor Who episode, The Doctor's Daughter, and by the end of the video, you'll have a better understanding of human genetics, reproduction, and development.
Everyone gets processed.
Oh, it's taking a tissue sample. Ow. I'm extrapolating here. Some kind of accelerator?
Are you alright?
No. It's just...
Arm yourself.
Where did she come from?
From me.
From you? How? Who is she?
Well, she's my daughter.
Hello, Dad. Do you say daughter?
Technically.
Technically how?
Progenation, reproduction from a single organism. Means one parent is biological mother and father. You take a sample of diploid cells, split them into haploids, then recombine them in a different arrangement and grow.
There's a lot there, so let's dive right into some human biology. Now wait, you may be thinking, the Doctor isn't human though. Yes, true, but the rest of the people using this machine are human, so we can infer that it is built with human biology in mind. Therefore, when discussing the functioning of the machine, I will do so within the context of human biology. To start, let's break this down into steps. First, the machine would take a sample of diploid cells from the epithelial or skin tissue on his hand. A diploid cell is a cell with two full sets of chromosomes, one inherited from the mother and the other from the father. Then it splits them into haploid cells. A haploid cell is a cell with one set of chromosomes. Every cell in the human body is diploid except for gametes. Gametes are the sperm in males and eggs in females used for reproduction. To split a diploid cell into a haploid cell, the diploid cell would undergo meiosis. Epithelial cells do not undergo meiosis. Only germ cells like spermatocytes in males and oocytes in females can create gametes. So first, the DNA would have to be extracted from the epithelial cell and inserted into an enucleated germ cell. This process is similar to a cloning process that has been done before, so it's plausible. These germ cells would then undergo meiosis, finally creating the haploid gametes. The process of meiosis includes a couple ways that create genetic variation, including crossing over, in which genetic information between homologous pairs is swapped, and the randomization of which chromosome ends up in which daughter cell. These two processes together create infinite possibilities for genetically unique gametes. The machine would then recombine them, creating a zygote through fertilization. The genetic material would all be from one parent, but because meiosis randomizes the genetic information in each gamete, different combinations of alleles would create different expressed traits within the offspring. Therefore, this machine could create a genetically unique offspring from the parent. This being said, it really shouldn't, for the same reason identical twins should not reproduce were that biologically possible. The goal of sexual reproduction is introducing genetic variation into a population. When genetically similar individuals, like relatives, reproduce, they could create a unique offspring, but recessive genetic disorders, mutations, and conditions would be much more likely to be expressed because both parents would be more likely to have and pass on that recessive allele. This machine would likely have complications with creating a viable zygote and would be completely unsustainable for multiple generations. Now that the machine has created a genetically unique zygote, the next step is to grow very quickly. A common misconception about cloning is that the clone would automatically be the same age as the person who is cloned. In reality, the clone will start out as an embryo and grow up at the same rate as anyone else. Although the Doctor's daughter is not a clone because she isn't genetically identical to the Doctor, the same rules apply. Cells have biological regulators limiting the rate of cell division, one of your body's defenses against cells becoming cancerous. Also, many parts of growth rely on environmental factors of being raised and nurtured, such as brain and muscle development. The growth of an entire human being cannot be sped up into a matter of minutes. Tissue development takes time, especially bone ossification. If you've ever broken a bone, you know it takes weeks for your cells to regenerate that bone tissue. To form a whole skeleton in seconds, not possible. So, in short, there's no possible way to grow a young adult this quickly. Basically, yes, you could create a viable zygote using this method, but it wouldn't be healthy, ethical, or sustainable. And with this technology, there's a significantly better alternative, sampling and combining DNA from two different parents. However, you could not grow that zygote into a healthy young adult in seconds.